Hi folks, today we're talking about chapter 21, nucleic acids. So today we're going to be talking all about DNA and RNA, nucleic acids, the bases, all that kind of fun stuff. So nucleic acids are involved in the transfer of genetic information from an existing cell to a new one. Most of us are probably aware of this in terms of people and the growth of cells for humans, but this can be at any level, whether it be the DNA of a goat or a fruit. All of these contain nucleic acids, which contain the genetic information to help the next cell carry on the traits. So when we're looking at nucleic acids, the bits and pieces of it will contain nucleotides. Those nucleotides will be made up of either pyrimidines or purines, a ribose or deoxyribose, and phosphates. So we got to think back all the way to our sugars chapter, where we started learning about ribose and deoxyribose. Technically, deoxyribose is not a carbohydrate because it does not follow the same pattern because of the fact that it is missing this oxygen. The structural difference is pretty easy. At the 2-carbon, ribose, the sugar, loses one of its hydroxyl groups to become deoxyribose. Both are still in beta conformation. When we have the difference between a pyrimidine and a purine is going to be these N-cyclic systems. So both are ring systems that will contain nitrogen. In fact, both of them have these aromatic nitrogen-containing 6-membered rings. However, purine will have an additional 5-membered ring on it. When we're looking at the nucleotide bases, they can be split into 2 categories, the purines and the pyrimidines. Within the purines, we have adenine and guanine. Again, remember, here's that 6-membered ring, here's that 5-membered ring, and then they are decorated differently. In the pyrimidines, we have cytosine, and then we have 2 other pyrimidines that are unique either to RNA or DNA. Uracil, which is in RNA, and thymine, which is in DNA. Remember, as we're going through and we're using these sugars, the DNA or the deoxyribose or the ribose, they're always going to be numbered, right? We can go from the anomeric carbon at the 1 to the 2, 3, 4, 5. So when we're adding those purine and pyrimidine bases, we're always going to attach them on the 1 position of the sugar, and the phosphate will always add to the 5 position of the sugar. So here we have a adenine being added to the 1 position of the sugar ribose, and the phosphate group being added to the 5. The nucleic acids will have a sugar as part of them, either ribose or deoxyribose, and a nitrogen-containing base, either pyrimidine or purine, bonded at the 5 position, and in addition to that, the phosphate group, the nitrogen-containing base will be at the 1 position, and the phosphate group will be at the 5 position. So while it may look something like this, sometimes we represent it in a very simple box structure as we see here. Now, the primary structure of nucleic acids, just like when we were learning about proteins and we were doing primary, secondary, and tertiary structure there, we can use the same kind of idea when we're talking about nucleic acids. Nucleic acids will have a sugar phosphate backbone. This means that as we go along the edge, at the 5-carbon of one nucleotide will be joined to the 3-carbon of the next. So what do we mean by that? Here's the 5-carbon, right? This is the one that has the phosphate. So sugar, phosphate. The next one in the loop would be another sugar, then phosphate, then sugar, then phosphate. And then this is the 3 position, this is the 5 position. So off of here, if this was more of a backbone, this would be a phosphate group here, which would be attached to the 5 position of another sugar down here. The linkage between each of these bonds is a phosphodiester. So just like we've had before with esters, we're going to have that linkage, but it's a phosphate diester. So here we can see that 1,3 addition, here, or excuse me, 5,3 addition. Phosphate group going to the 5 carbon of the sugar. So we go phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, 5, 3, 5, 3, 5, 3, 5, 3. And then at each anomeric carbon, at the 1 position, we have one of our nitrogen-containing bases. The structure of DNA is what we call a double helix for its secondary structure. And when we think of a double helix, probably the easiest way to think about this would be if you had a ladder and you twisted it. The sides of the ladder are going to be that sugar phosphate backbone. Each side of the ladder would be that sugar phosphate, sugar phosphate, sugar phosphate going 5, 3, 5, 3, 5, 3. And then the rungs of the ladder, or the steps of the ladder, would be the bases. Now, the bases will end up having hydrogen bonding interactions occurring between the adenine and the thionine and the guanine and cytosine. Some of them only have 2 hydrogen bond interactions, some of them have 3. So here you can see an example of both of those. As we can see, the adenine and thymine, they have hydrogen bonding interaction at these 2 locations and have 2 of them, where the guanine and cytosine have 3. The chromosomal packing of DNA. So a human cell contains 46 different chromosomes. Each of these chromosomes contain 1 molecule of DNA and histones. Histones are like little basic proteins that kind of help organize the coiling up of the DNA so it doesn't become clustered. DNA contains segments called genes, and each gene will direct the synthesis of a specific protein. In total, a human cell contains approximately 25,000 genes. So we know that in each cell we have the DNA, the genes, the directions for the synthesis of each protein in the human body. So when a cell is going to replicate, it needs to copy all of this DNA. To make an exact copy of the DNA, it needs to get passed to the new cell correctly. And so there are a lot of safeguards in place to make sure that we have as minimal errors in the DNA replication as possible. DNA replication is what we call semi-conservative. This means that if we have a parent DNA molecule, this parent DNA molecule will split, and a daughter strand will be made and will pair up with each parent strand. So each of the new daughter strands that are made has 1/2 of the original parent strand. What this allows it to do is kind of help with the errors, make sure that minimal errors are occurring during replication. So this is another example of that. Blue representing the parent strand. During replication, we now have 2 daughter strands, which are partially new and partially old. Now the DNA as it occurs will happen at multiple spots along the DNA all at once. So DNA does not start at one end and go all the way down. What ends up happening is we have multiple little segments that occur all at once and then spread out from there until the entire strand is completed. This is a much quicker process than starting at one end and unzipping the whole way down. When this is done, the DNA is unwound by DNA helicase, and we form what we call a replication fork. It kind of looks like a tuning fork, and that's where it gets its name. DNA segments, called Okazaki fragments, are synthesized by DNA polymerase between each of the forks. So these Okazaki fragments here, DNA can only be done in one direction. It can only go from 5 to 3. That's just how the protein works. So on one side, we're able to go from 5 to 3 very easily. However, on the other side, we have to go 5 to 3 in sections, and then they will be knitted together because it's going to unwind this way. So it's going to make 5 to 3, and it's going 5 to 3, 5 to 3. It's very easy to do it this way. So where these nicks are, these will have to be fixed over later. As we said, replication will occur simultaneously at many points along the DNA. It would take days and days and days for something as simple as a fruit fly to replicate DNA if it was to be done over a full strain. If it went from one end to the other, it would be 16 days. However, when it's done in these small sections, it takes about less than 3 minutes because of the fact that we have multiple replication forks going simultaneously. We have, you may have heard of the term PCR. Lately with COVID, as one of the way we do testing, we are doing DNA testing to replicate and grow a sample to see if there is COVID DNA in a PCR test. And so PCR stands for polymerase chain reaction, and it mimics the natural process of DNA replication so that we can do it on a lab bench. Small quantities of DNA are then mixed and added with DNA polymerase, some magnesium chloride, a few other factors, including the building blocks needed to allow the daughter strands to be formed. It is heated to unravel the DNA, cooled down to allow the primers and the hydrogen bonds to be separated in the strands of the DNA, and then heated at a much more physiological acceptable temperature to instigate polymerase to occur. DNA polymerase is, PCR is used for a number of techniques, including things like genetic disease testing. Sometimes we do it to identify bacterial or viral infections. It is pretty fast in terms of a scientific technique, straightforward to use. Sometimes it can be a little more pricey than other types of testing. However, when you are using something as time sensitive as, for example, a COVID test, it is a good trade-off. RNA is similar to DNA, but again, we know that RNA is going to use ribose instead of deoxyribose. But there are some other differences as well. Unlike DNA, RNA will be single-stranded. It's long, it's unbranched, is joined 3 to 5, just like the DNA is. However, it tends to be made of smaller nucleotides, smaller units of it. We find it in many different areas, including the nucleus, the cytoplasm, mitochondria, and that is because they have different roles. RNA comes in 3 flavors: messenger RNA, ribosomal RNA, and transfer RNA. And these different types of RNA exist all over the cell because of the fact that they have different jobs. So how is RNA different from DNA? Again, we know that we have ribose versus deoxyribose. If you were paying attention earlier, we have uracil as a base instead of thiamine. And because they are single-stranded, we don't necessarily have the same one-to-one ratio that we do with DNA when it comes to the complementary bases. It does have regions where it'll be double helical or looped in its structure, but these loops are why we don't have a one-to-one ratio. So in the areas where it does overlap and do a double helical structure, we do have base pairing going on, just like in DNA. But these loops mean that the ratios can be completely off. We could have one entire loop that is simply just uracil, which would obviously then have a huge effect on the ratio of uracil to adenine as we were looking through the overall RNA structure. So in terms of different forms of RNA and where they exist, this is done in E. coli as a very simple a kind of comparison. But ribosomal RNA would make up about 80% of the RNA in the cell. Transfer RNA about 15, and mRNA or messenger RNA making up about 5. And as you can see, we have a huge variety in the types of sizes of these nucleotides of RNA. So what do each type of RNA do? Messenger RNA will carry genetic information from DNA to the nucleus site where a protein is being made, right? It has a very short lifespan. Its entire job is to take the genetic information to the cytoplasm. Once that's done, it is recycled and used for something else. Ribosomal RNA is the main component of a ribosome, and this is where the protein is being made. So most of the RNA is going to be ribosomal RNA because that is the workhorse of the protein synthesis. Lastly is transfer RNA. tRNA is the mode by which amino acids are delivered to the protein synthesis site. So tRNA will deliver amino acids to the ribosomal RNA, where it will be knitted together to make a protein. Each type of tRNA is specific to one amino acid. Therefore, cells contain at least one specific type of tRNA for each common amino acid. They're very, very small in comparison to other types of proteins that we've looked at, being around under 100 nucleotides long in length. There are two important regions of tRNA. The anticodon, which allows the tRNA to bind to the mRNA, right? And this will allow the mRNA to tell the tRNA which amino acid to pick up. And then we also have the side of it that will bind to the amino acid ester and bring it through transport to the protein synthesis site. So here's a structure of tRNA. You can understand why it's called tRNA. It's shaped like a T, and there are two different sites on it. We have the 3' side where we have the amino acid attachment, and then we have the codon site attachment. This is where the tRNA would attach to the mRNA to make sure that we're getting the right amino acid. So what will end up happening is we have our 3' side here on our sugar. Right, here's our sugar phosphate backbone. Here is our new acid that we're bringing in, and it will be done by the tRNA. So the flow of genetic information, if you've taken a biology class before, you've probably heard of this central dogma before, but basically it's just the process by which DNA gets turned into a protein. So DNA can go through a replication, and this would happen with each new cell. So DNA would replicate itself, right? Those tuning forks going through the Okazaki fragments, going from the 3 to 5' side, and knitting those together, and then having two new strands of DNA, each containing an original parent strand as well as a new strand. So replication is always occurring. At some point, we might need to then go make a protein. So going from DNA to RNA, we have transcription. The way I remember DNA to RNA is transcription is there's an extra R in transcription. DNA is then translated into a protein, DNA then converts into RNA, and RNA then converts into a protein through translation. DNA being transcribed into mRNA. How does this process work? Well, again, we're going to have some sort of protein to make the RNA. What happens is part of whatever sequence of DNA we need will unwind, and we'll have this single strand exposed. The mRNA will be transcribed using the appropriate bases to create the mRNA. The DNA strand will then rewind, and the mRNA is then synthesized. Transcription, right? So going from DNA to RNA produces heterogeneous nuclear RNA. What we mean by this is it contains information that is both useful and not so useful. We have things called introns and extrons in DNA. An intron DNA segment carries no code for an amino acid. So for going from DNA to RNA, it is not useful information. An exon is what carries a code. So the way I like to remember this is that an intron would stay inside the cell, an extron would leave the cell to go make a protein. hnRNA stands for this heterogeneous RNA, which contains both the introns and extrons. However, for it to become mRNA and be useful for coding for amino acids to build a protein, we need to cut out all the introns and have all the exons knitted back together. So here's an example of hnRNA with both the introns and extrons in there, and those need to be cut out during transcription and knitted back together as just the exons. The mRNA is the template from which the amino acids are going to be assembled to make our protein. It's an exact, what we say, quote unquote, word-to-word translation, which means that for each codon, it's calling out a specific amino acid. The codon sequence for 3 nucleotide bases that represent a code word for the mRNA molecules. So here's an example of those codons. So they're all 3 units long. Some amino acids have more than one codon that can represent them, and some only have one. Especially methionine. Methionine is very important because this is going to always be the initiation step. So all of them will start with methionine and then go from there. So codons are going to be a 3-letter word, or excuse me, a word, quote unquote, but 3 base pairs knitted together to call out a specific amino acid. The codes can be degenerate, which means we have multiple codes all representing the same amino acid, or they can be precise, where it would only be one. There's always going to be a code to initiate the strand being made, and there's a code for ending the strand. And then, interestingly enough, something like the code GCA for alanine is present in almost all organisms. So almost all organisms use the same code for alanine. So how does this work? Again, remember, we have mRNA, this subunit here, this whole thing here, this is basically going to be our rRNA, our ribosomal RNA, and here's our tRNA bringing in our amino acids. So what will happen is mRNA and a small ribosomal unit will join, and this will initiate a codon. AUG standing for methionine, right, that initial codon. The large ribosomal unit will come on top, and then what will end up happening is as each tRNA is brought, it will link into the large ribosomal unit, and it will start knitting together the different amino acids as they come. So we had methionine, and then next we have something for phenylalanine, and the next one after that is going to be valine, and each time we're going to grow and grow. Here's that primary structure of our protein growing. So this is a little more abstract, but here we can see, right, our small subunit and our large subunit kind of pinching together to read as it goes along the mRNA from the 5 to the 3. And as it goes along, the first primary structure of the protein begins to form. As it gets long enough, secondary structure will actually start to be done as well, even while it's still growing. At some point, there will be a termination, and it will release the completed protein. Now, with all these safety features in place to help minimize changes and errors in the DNA, sometimes those do occur. These are called mutations. This is any change that results in an incorrect base on the sequence of the DNA. They do occur naturally. DNA replication is not perfect. However, they can be induced by environmental, including radiation from UV, X-rays, things like that, or chemical agents. Not all mutations are necessarily harmful or even something that would be noticeable in terms of, for good or for bad. So we can utilize DNA to help do some genetic engineering. This has been done for a long time, especially with things where we need to have medical applications of it. So recombinant DNA uses restriction enzymes to cleave specific parts of DNA where we want to either, produce a specific protein. For example, we can use this as a way to grow insulin. Human insulin that is used for the purposes of treatments of diabetes is not produced by humans and cultivated that way. We actually utilize either E. coli. Previously, it was done in cows to harvest human insulin. So what we can do is insert portions of DNA into something like an E. coli, which is very easy to keep alive and feed, and they grow very rapidly, and let them reproduce the protein over and over again. So in this case here we have a bacterium and its plasmids. We isolate one of them, we cut it, and we insert the portion of human DNA that is the desirable genes in there. So let's say these contain the genes to produce the protein of human insulin. They're spliced back together, and now we have a recombinant DNA that is both E. coli and human. And this is put back into the cell, and the cell is allowed to go grow as it normally would and propagate. And what this will do is, what this will do is allow the bacterium to grow and produce the proteins for us, which can then be isolated and packaged and used as medical treatments. So this is the end of chapter 21. We'll be moving on to chapter 22, but I'm going to stop the video here for this unit, and you can pick up with chapter 22 next.